In this session, let us discuss about ribosomes. Ribosomes are ribonucleoprotein particles present in prokaryotic and eukaryotic cells. These are organelles which do protein synthesis, and these are the organelles which are present in both prokaryotes and eukaryotes. They are also called protein factories. They are found freely in cytoplasm as well as attached to the endoplasmic reticulum. And when ribosomes are attached to endoplasmic reticulum are called rough endoplasmic reticulum. Ribosomes are first observed by Cludi in 1941 and they named it as microsomes. The term ribosome was first coined by Pallade in 1955. Ramakrishnan, States and Ada E. Inath got Nobel Prize in 2009 for studying the structure and function of ribosomes. These organelles are also observed within chloroplast and mitochondria where they do protein synthesis. Prokaryotic ribosomes are having 70S with subunits 30S and 50S, whereas eukaryotic ribosomes are having 80S ribosome with 40S and 60S subunits. Fungal mitochondrial ribosomes are of 77S type. The S associated with ribosome stands for Swedberg unit, which is the sedimentation coefficient of each unit. Animal cell mitochondrial ribosomes are having a size of 60S, and the number of ribosomes present in a cell is directly related to RNA content of the cell. Now let's see the structure of ribosomes. Ribosomes are spherical in shape. Often their size remains constant in an organism or in the cells of an organism. The prokaryotic ribosomes are smaller, while in eukaryote they are larger. The size of prokaryotic ribosome is 150 angstrom, it's the diameter, whereas the eukaryotic ribosomes have a diameter of 250 angstrom. The ribosomes are formed of two subunits, the larger subunit and the smaller subunit. This is the smaller subunit and this is the larger subunit. When they are not doing protein synthesis or when they are not functional, this unit occurs separately in cytoplasm. They will be uniting together only at the time of protein synthesis and especially in the presence of messenger RNA. Messenger RNA carry genetic information from nucleus to ribosome. During protein synthesis, multiple ribosomes are seen attached to a single messenger RNA. During protein synthesis, multiple ribosomes are attached on a single messenger RNA, and this condition is called polyribosomes. Such a string of ribosome is called polyribosome or polysome. During the association of messenger RNA with ribosome, when protein synthesis is happening, the smaller subunit holds the messenger RNA. A functional prokaryotic ribosome has three binding sites, an A site, a P site and an E site. A site, P site and E site. A site carries transfer RNA activated with an amino acid, or A site carries tRNA containing activated amino acid. The P site carries a transfer RNA with a polypeptide chain. This is A site, it carries an amino acyl tRNA which is activated, and the P site carries a growing polypeptide chain, and E site is otherwise called exit site, through which the deacylated tRNA, after giving the amino acid for protein synthesis, the deacylated tRNA is released, but an eukaryotic ribosome has only two sites, an A site and a P site. Whether it is prokaryote or eukaryote, the binding sites are contributed or formed by the contribution of both subunits. The smaller subunit is flat and discoid. This is the smaller subunit. It is flat and discoid with a lower surface slightly convex and upper surface slightly concave. And thus the smaller subunit doesn't have a symmetry, so it is asymmetrical. And it is constricted at the middle of the flat body. Middle there is a constriction is present. There is a cleft on the upper surface of the smaller subunit which divide the subunit into a head and a base. And this is the cleft which divide the subunit into a head region and a base region. Whereas the larger subunit is spherical structure with 3 convex sides and a concave side. It have 3 convex side and then there is a concave side at the bottom. There is a large protuberance from one side of the rim of the lower surface. On either side of the protuberance there is a depression. And this is the protuberance and this is the depression. Due to depression, a clear stalk is formed on one side of the protuberance in larger subunit. During union, the concave surface of the smaller subunit is bound to the bottom of the larger subunit. This is how the union of ribosome is happening. The protuberance of the large subunit is aligned with head of the smaller subunit. When this union is happening, 2 spaces are formed between the subunits and they are the A site and P site. A site carries tRNA containing activated amino acid, whereas P site carries tRNA containing peptide chain. Coming to the chemical composition of ribosomes, ribosome contains RNA, proteins, enzymes, metal ions, and hence they are called ribonucleoprotein particles. In eukaryotic cell, ribosomal RNA are found in 4 forms. The larger subunit of eukaryote contain 28S RNA, 5S rRNA, 5.8S rRNA, whereas the smaller subunit contain 18S rRNA. In the case of prokaryotes, the larger subunit contain 23S rRNA and 16S rRNA, and the smaller subunit of prokaryote is 5S rRNA. The 70S ribosomes contain 50s to 60 proteins, while the 80S ribosome contains 70 to 80 proteins. The ribosomal proteins are classified as core proteins and split proteins. The enzyme factors like initiation factor, elongation factor and termination factor are present in ribosome. Ribosome also contain metal ions like magnesium, calcium, manganese, iron, etc. Coming to origin of ribosome, ribosome is having a biogenesis. In eukaryotes, ribosomes are synthesized in nucleolus and transported to cytoplasm. In prokaryotes, they are synthesized in protoplasm. In eukaryotes, the chromosome which is having a nucleolar organization region, ribosomal rRNAs will be transcribed. Initially formed rRNA will be a 45S rRNA and it will be methylated. Later, this 45S rRNA will be cleaved at the regions where it is non-methylated and the individual ribosomal rRNA molecules are formed. So, from the 45S rRNA, by cleaving at non-methylated site, the smaller rRNA molecules are formed. The ribosomal proteins will be synthesized in cytoplasm and transported to the nucleus. So, the rRNA synthesis is happening within the nucleus. The ribosomal proteins will be synthesized in cytoplasm and transported to the nucleus. The 60S and 40S ribosomal subunits will be formed within the nucleus by the combination of specific rRNAs and ribosomal proteins. Once formed, these subunits will be released to cytoplasm through the nuclear pore. In cytoplasm, these subunits will be united to form a functional ribosome only during protein synthesis in the presence of messenger RNA. Coming to the function of ribosome, all functions are associated with protein synthesis. During protein synthesis, a messenger RNA will be produced from the genomic DNA, and this messenger RNA will be carried to the cytoplasm. It will undergo splicing and processing, and the functional messenger RNA will be there in the cytoplasm. In the presence of functional messenger RNA, the ribosome unit will be united together. Now protein synthesis will start. The protein synthesis process is carried out by ribosome. Initially, the ribosome has to read the message on the messenger RNA and decode it. This is carried out by ribosome. After reading the message, correct amino acid acyl tRNA has to be identified and accepted. This process is carried out by the ribosomes. Once the correct amino acids are received, the next amino acid will be attached to the amino acid by peptide bond formation. This is happening within ribosome. And after the release of amino acid by tRNA to the ribosome, the deacylated tRNA is released by ribosomes. So all the functions of ribosomes are associated with protein synthesis. They do protein synthesis, they do decoding of messenger RNA, they do accept the correct amino acid tRNA, they do the peptide bond formation, they release the deacylated tRNA. Thanks for your listening.